Nucleic acids are polynucleotides, which is poly is mean many, that is long chain-like molecules composed of a series of nearly identical building blocks called nucleotides. Each nucleotide is made of three parts: a nitrogen-containing ring structure called a nitrogenous base, a 5-carbon sugar, and at least one phosphate group. The sugar molecule has a central position in the nucleotide, with the base attached to one of its carbon and the phosphate group attached to another. The nitrogenous bases of nucleotides are organic, which is carbon-based molecules made out of nitrogen-containing ring structure. Why called bases? Because some of nitrogens in a base can be protonated, which is P-carb and extra H+ ion. Nitrogenous bases decrease the hydrogen ion concentration in a solution and adduce bases in the acid-base sense. Types of nitrogenous bases are adenine, guanine, cytosine, thymine, and uracil. The structure of nucleic acid. Nucleic acid is a very large molecule that have two main parts. The backbone of nucleic acid is made of alternating sugar and phosphate molecules bonded together in a long chain phosphodiester bond. Each of the sugar group in the backbone is attached to a third of molecule called as nucleotide base. A consequence of the structure of nucleotide is that a polynucleotide chain has directionality. That is, it has two ends that are different from each other. The phosphodiester bond links the 3' carbon in the sugar ring of one nucleotide to the 5 carbon on the next nucleotide. What is the function of nucleic acid? The function of nucleic acids are store information of every living cell, which is the genetic material. Then, transmit and express that information inside and outside the cell nucleus to the interior operation of the cell. For example, nucleic acid carry genetic information which is read in the cells to make the RNA and proteins by which living things function. The well-known structure of the DNA double helix allows this information to be copied and passed on the next generation.
Purines and pyrimidine. Purines and pyrimidine are two families of nitrogenous bases that make up nucleic acid. In other words, they are building blocks of DNA and RNA. While they are similar in many respects, there are a number of key differences between them. Purine are adenine and guanine. They are both composed of a double ring structure and larger than pyrimidine. Both of these occur in both DNA and RNA only. There are three main types of pyrimidine. However, only one of them exists in both DNA and RNA, which is the cytosine. The other two are uracil, which is RNA exclusive, and thymine, which is DNA exclusive. These all have similar ring structure, and purine will always bind with pyrimidine, which is known as the complementary pairing. The purine on one strand of DNA will form hydrogen bond with the corresponding pyrimidines on the opposite strand of DNA and vice versa to hold the two strands together. Adenine will always pair with thymine and guanine will always pair with cytosine in DNA. And adenine will pair with uracil and cytosine will pair with guanine if RNA is involved.
Is DNA. The definition of DNA is the abbreviation of deoxyribonucleic acid, organic chemical of complete molecular structure that is found in all prokaryotic and eukaryotic cells and in many viruses. DNA codes genetic information for the transmission of inherited traits and instruction for protein synthesis. DNA is a key part of reproduction in which genetic heredity occurs through the passing down of DNA from parent or parent to offspring. Cell DNA is organized into dense protein DNA complexes called chromosome. In eukaryote, the chromosomes are located in the nucleus, although DNA also found in mitochondria and chloroplast. Prokaryotes, which do not have a membrane-bound nucleus, the DNA is found as a single circular chromosome in the cytoplasm. Some prokaryotes, such as bacteria, and a few eukaryotes, has extra chromosomal DNA known as plasmid, which are autonomous, self-replicating genetic material. Plasmid has been used extensively in recombinant DNA technology to study gene expression. Discovered the structure of DNA. The discovery of DNA double helix structure is credited to the researcher James Watson and Francis Crick, who, with fellow researcher Maureen Wilkin, received the Nobel Prize in 1962 for their work. Many believe that Rosalind Franklin should also be given credit since she made the revelatory photo for the DNA double helix structure, which was used as evidence without her permission.
What is DNA made of? Each strand of a DNA molecule is composed of a long chain of monomer nucleotides. The nucleotides of DNA consist of a deoxyribose sugar molecule to which is attached a phosphate group and one of 4 nitrogenous bases, which are 2 purines, adenine and guanine, and 2 pyrimidines, cytosine and thymine. The nucleotides are joined together by covalent bonds between the phosphate of one nucleotide and the sugar of the next, forming a phosphate-sugar backbone from which the nitrogenous bases protrude. One strand is held to another by hydrogen bonds between the bases. The sequencing of this bonding is specific. Example, adenine bonds only with thymine and cytosine only with guanine. How is DNA replicated? Replication occurs in 3 major steps, which are the opening of the double helix and separation of DNA strands, the priming of the template strand, and the assembly of the new DNA segment. During separation, the 2 strands of DNA double helix uncoil at a specific location called the origin. Several enzymes and proteins then work together to prepare or prime the strand for duplication. Finally, a special enzyme called DNA polymerase organizes the assembly of the new DNA strand. The following description of this 3-stage process applies generally to all cells, but specific variations within the process may occur depending on the organism and cell type. Can you edit DNA? Gene editing today is mostly done through a technique called clustered regularly interspaced short palindromic repeats (CRISPR), adapted from a bacterial mechanism that can cut out specific sections in DNA. One use of CRISPR is the creation of genetically modified organism (GMO) crops.
RNA is typically single-stranded and is made of ribonucleotides that are linked by phosphodiester bonds. A ribonucleotide in the RNA chain contains ribose, which is the pentose sugar, one of the 4 nitrogenous bases, adenine, uracil, guanine, and cytosine, and a phosphate group. The subtle structural difference between the sugars gives DNA added stability, making DNA more suitable for the storage of genetic information, whereas the relative instability of RNA makes it more suitable for its short-term function. The RNA-specific pyrimidine use uracil instead of thymine to form a complementary base pair with adenine. Even though RNA is single-stranded, most types of RNA molecules show extensive intramolecular base pair between complementary sequences within the RNA strand, creating a predictable 3D structure essential for its function. There are 3 types of RNA. The first one is mRNA, second tRNA, third rRNA. The function of mRNA is to carry the message of transcript DNA codes or polypeptides from the nucleus to the ribosomes. The function of tRNA is to carry specific amino acid to the ribosome, aiding to translation. The function of rRNA is to associate with specific proteins to form ribosomes.
Transcription. We could say like this: when we transcribe information, we use the same language to write it down. Like in a class, when the teacher asks you to read the transcript, you speak the same language as written. Translation is exactly what it says. We are changing the language. RNA translation changes the language from nucleotides to amino acid. RNA is synthesized by RNA polymerase. So what is the role of each eukaryotic RNA polymerase? RNA polymerase 1 will synthesize most rRNA. RNA polymerase 2 will synthesize mRNA. And lastly, polymerase 3 will synthesize tRNA and some rRNA. When starting transcription, where does RNA polymerase bind? It will bind to Tata box, which is located within the promoter region of a relevant gene at about -25. Transcription will continue along the DNA coding region until the RNA polymerase reach a termination sequence on stop signal, which results in the termination of transcription. The DNA double helix reforms. The primary transcript is termed hnRNA. mRNA is derived from hnRNA via post-transcriptional modifications. What are three major post-transcriptional modifications that turn hnRNA into mature mRNA? The first stage is splicing by removing introns and joining of exons. The second step is 5-cap, which is addition of a 7-methylguanylate triphosphate cap to the 5' end of the transcript. The last step is 3-polyethyl, which is addition of adenosine bases to the 3' end to protect against degradation. Once mRNA transcript, it can exit the nucleus through nuclear pores, and then translation will take place. Translation occurs in 3 steps, which are initiation, elongation, and termination. During the process translation, mRNA will transcript into a functional protein. Thank you for watching and good luck for your final exam.